All right. Hi guys. I think I know most of you. I'm Salman, for those who I don't know. I'm a fourth year, and I am taking a year off to do research under Dr. Tom Liebman, studying trachoma in countries in Africa. And so the title of my project is called Challenges to the WHO Strategy for Trachoma Elimination as a Public Health Concern. So I'm just going to start off talking about what kind of trachoma is as a disease. It's not something we're too familiar with, it's not something we learn about in med school. And then I'll kind of talk about the epidemiology, the WHO's campaign for elimination, and then we'll get into like my research question and the methods that I'm going to be using to study this. So this is a human epithelial cell, and what you can see if this laser pointer had worked was that this is an inclusion body that contains chlamydia. And so trachoma is an infectious disease caused by, the causative agent is chlamydia trachomatis. And if you guys remember from I3, it's intracellular and it's a bacteria. Those are really the only important things we need to know. Because it's a bacteria, it's susceptible to antibiotics. And that becomes important later. So just going to go through the pathogenesis very quickly. So like I mentioned, trachoma is an infectious disease, and so it affects the eye, and what happens is you get ocular infection with chlamydia, and that causes an inflammatory response, and then usually you clear that infection, and that happens in children. Children are the people who are getting infected, and then they clear the infection, but because you only develop partial immunity to this disease, you get reinfected really quite easily. And so you get repeated bouts of infection and reinfection and inflammation and reinflammation. And over time, that causes scarring and a series of sequelae that we'll talk about a little bit, in just a little bit with pictures. And those sequelae happen in adults. So just for the basics, infection happens in kids and the sequelae happen in adults. So I'm going to show a couple of pictures. They're kind of gross, so just as a forewarning. Show in a second. Yeah. So this is just a prevalence curve. On the y-axis we have prevalence of infection. X-axis we have age. And the blue curve shows the prevalence of infection, and the purple curve shows the prevalence of scarring. And so what you can see is the infection happens in kids, mostly school-age children, and scarring happens like much later in life. And that's kind of all you need to know. So this is some slides that I got from my PI. I'll have my own slides coming up. I'm taking a trip to Ethiopia in 2 weeks. Really excited about that. And so for my next presentation, I'll have pictures for you guys. This is the inside of Tom's eyelid. And so as you can see, a normal healthy eyelid has blood vessels going up and blood vessels coming down. You get this watershed area in the middle. And that's what it's supposed to look like. This is an eyelid with trachoma. What you see is the normal blood vessels are all gone. You get a lot of redness, a lot of inflammation, and you start to see these little white circles appear, and these are follicles. Just like you get follicles in your lymph nodes, secondary germinal centers, that's exactly what's happening here. And if you also notice, you see this fly here. So this is Muscus sorbens, and it's a face fly. And what it does is it feeds on the ocular and nasal secretions of children. And it's thought to also play a role in transmitting infection, because it'll, like by chance, carry secretions from an infected child on its legs and then land somewhere else and transmit that infection. But mostly, infection is thought to be transmitted from direct hand-to-eye or hand-to-hand contact between infected individuals. So this is the next stage of trachoma, which we saw here. This is just inflammation and development of follicles. Here what we're seeing is more of the follicles, these white things here, but if you look really carefully, this kind of white fibrous area is the beginnings of a scar. And so that's when the scarring starts to form. And this is a little bit later, this is a lot of scars. So the kind of networks you're seeing of white tissue, those are fibrous scars. And as the scar forms and the fibrous tissue is forming, it causes contracture of the tissue. and it causes the eyelid to turn inward, and that's called entropion. And then because the eyelid is turning inward, the eyelash is turning inward and scratching the eyeball, and that's called trichiasis. And when the eyelash scratches the eyeball, it causes a corneal ulcer and corneal opacity. And here we can see the cornea is opaque, and it's here you've progressed to the stage of irreversible blindness. And so what we try to do is target these individuals early in the stage of the disease when they have active trachoma and inflammation to kind of prevent them from getting to this point. What's interesting is at this stage, which is trichiasis, before they've become blind, you can do a surgery, bilamellar tarsal rotation, which is done in the field by trained nurses, and they flip the eyelid right side up, which is pretty interesting. I saw a video of it on YouTube. If you're interested in doing that. So this is just kind of to show the 4 stages. So you get inflammation, you get more inflammation, and that kind of goes on. These top 2 pictures happen in kids a lot, and then over time, as you get older and become an adult, that process causes scarring, which you see in the bottom left-hand picture, and then the bottom right-hand picture, it's when you've already developed a corneal opacity and you've become blind. So this is an interesting picture. This is taken from Ellis Island in the early 1900s, and what you see here are immigrants coming into Ellis Island and government immigration workers flipping the eyelids of people and checking to see if they have trachoma. And if they do, they either got sent back or put into quarantine. Prior to the late 1970s, 1980s, trachoma was seen not just in developing nations around the world, but also in North America and Europe, particularly Eastern Europe, which is where a lot of these immigrants were coming from. And so it was kind of a global problem. Today, this is what the map of trachoma looks like. You see that it's pretty much gone from North America and a lot of Europe. And it's highly endemic in areas in Africa and Southeast Asia. And now it's thought to exist endemically in about 60 countries. Yeah?
How was it eradicated from the US?
So the last known case in the US was, I think, 1978, and it's thought to be mostly due to better kind of, like developing nations have better living conditions, or developed nations, sorry, have better living conditions. We'll talk a little bit in a couple of slides about how the WHO has been implementing a strategy to try to eliminate it, and part of that strategy includes environmental improvements and facial cleanliness, and it's thought these things have helped to eradicate it from developing countries or developed countries. Yeah. So this is epidemiology. In 1995, the WHO published its first report on the global incidence of or prevalence of blindness, and trachoma was the #1 infectious cause of blindness. #2 overall. It had about 8 million people blind, and that was estimated, and 146 million people with active trachoma. Today, that prevalence has gone down significantly, with about 1.2 million people blind and 21.4 million cases of trachoma. In large part, it's thought that this decline in prevalence has been because of the WHO's program to eliminate trachoma. It's a multi-pronged approach, but the one prong that has been shown to be absolutely efficacious is the mass distribution of antibiotics, and that's kind of the focus of my research. So let's talk about mass distribution of antibiotics. First, let me just kind of explain why we need to mass distribute it to everyone. So you have, imagine these communities where you have endemic trachoma, something like 50% of school kids have it, and if you give only a certain number of kids the antibiotics, they'll clear the infection, but then they'll easily get reinfected by their schoolmates because people are in very close contact, and because of partial immunity, trachoma they just catch the disease over and over again. So you have to kind of give it to everyone. If there was a way that we could kind of look at, figure out exactly what kid has the infection and only give it to them, then you wouldn't need to do it with mass distribution, but there's not really a way to do that yet. So let's kind of talk about what the negatives would be of giving mass distribution of antibiotics. First, you have the side effects from the antibiotics. The antibiotic of choice that they give is azithromycin, and that has GI side effects. Not common, but you do see like nausea, diarrhea, vomiting, upset stomach. The other big thing that I'm sure all of you guys are wondering is, well, aren't you causing resistance? And it's a great question. And from what we know so far, we haven't seen chlamydia that's resistant to azithromycin. We are selecting for pneumococcal resistance, and that's been shown. However, those pneumococcal strains are, there's a couple things actually. First, once you stop giving the antibiotics, those resistant strains disappear. And then secondly, those pneumococcal strains are not resistant to the penicillins, which are the most widely used antibiotics in that region. And so we don't think resistance is a big problem, but we're starting to wonder if there are new strains that are being created, chlamydia strains that are resistant to azithromycin, and that's kind of what I'm going to be studying, which is how efficacious these antibiotics are. So next let's talk about the positives of giving antibiotics. First, it's the only proven intervention that works, and the good thing about azithromycin is that you only need to give one dose, and you only give it once a year, so it's really easy to distribute. And actually what we've shown, Tom's group, is that there's a mortality benefit. And they're starting a new trial called the Mordor study, which is going to be looking at whether these massive distributions actually have this mortality benefit for these regions that don't get healthcare. Yeah.
So with the single dose, do you guys still have like a bacterial load? 'Cause, in your graph you showed that like the line doesn't go down to 0. So are these people still like infected with like an intracellular bug that's like been dormant, essentially, or is it pretty much completely like cleared?
So the way that we test for whether or not someone is infected is that we swab their conjunctiva and then we PCR for chlamydial DNA.
Mm-hmm.
And so there's a couple reasons why if you give someone an antibiotic and they still test positive,
right.
Why that could be. It could be either that there's dead organisms, or it could be that the antibiotic wasn't 100% efficacious,
Mm-hmm.
which is what I'm studying. Like how efficacious are these antibiotics really? What's the percentage that if you give, or percentage chance that if you give someone an antibiotic, they'll clear the infection? So that's another. It's a great point. Yeah. Okay. So this brings me exactly to my research question. So I'll be looking at what is the efficacy of this antibiotic? Is the efficacy changing over time? And secondarily, I'll be looking at whether there's a loss of immunity. I'm not positive if I'll get to it today, but I might have time to talk about it. So here's a chart kind of describing to you, what I want to describe to you next is basically how I'm going to figure out what the efficacy of these antibiotics are. This is what the data looks like. I made it kind of simple in that I only included one mass treatment, and these red circles basically represent what the prevalence is at different time points. So on the y-axis you have prevalence, on the x-axis you have time, and then this green arrow represents the initiation of antibiotics. So baseline prevalence, for example, is 50%. At time t equals 0, we're going to give antibiotics and then measure the prevalence at subsequent time points thereafter. And so you might say, well, if this is your data, it's going to be really simple to measure what the efficacy is. All you have to do is measure this drop in prevalence, and you know that the drop in prevalence is proportional to the coverage, which is the percentage of people that you gave the antibiotics to, which we're measuring, at times the efficacy. So you just solve for a single variable. Turns out it's not that simple. This is what the graph actually looks like. The green circle represents what the prevalence of infection is truly right after you give the antibiotic, and everyone who's going to clear the infection clears the infection. It's a time point that we don't have in our data, we haven't measured it, and somehow we need to estimate it or figure out what it is, because the data just isn't there. And like I mentioned, people get reinfected so quickly that it's actually really quite hard to measure if you even try to think about what kind of experiment you could do to measure that time point. So that makes it kind of tough since we don't have that data. So this is the drop in prevalence that we want to know, and this is the drop in prevalence that we do know. So we somehow need to figure out what the green arrow is, and we only have the blue arrow. And that, here I only did one mass treatment. Imagine if this is what your data look like, which is 4 mass treatments. Then how are you going to estimate 4 time points if you don't even know what they are? So that's kind of the challenge that I'm presented with. And the way I'm going to solve it is with mathematical models. I want to apologize in advance strongly for what you're about to see. It's a little bit heavy in math, but I'm going to make it simple and not get too into the nitty-gritty. And I think it's pretty interesting. So what we're going to be doing is modeling how infection spreads through a community. I'm going to start with like a very, very simple model and then add components to it to the point where it kind of mirrors what's happening in real life. And then I'm going to use the data that we do have to solve for the unknown variables in this model, and that's the process called model fitting. Okay. So this is it. This is the world's simplest model. It starts by dividing the entire population into two compartments. You have people who are susceptible to infection, and you have people who are infected. So if you're in the community, you either exist in one of those two categories. We assume that there's no people being born, no people dying. We assume that there's no infection from outside. We assume that you don't develop immunity to the disease, which is what we see in the children that we're modeling. You know, people just get reinfected. So people move from being susceptible and not having the infection to becoming infected. And then once you're infected, you can go back to becoming susceptible again, either by recovering naturally from the disease or by getting antibiotics. But again, the devil's in the details as to how we are going to model this. So the rate at which you move from becoming susceptible to becoming infected is determined by this parameter called beta. And you're going to hear a lot more about beta in just a minute, but high level, what it represents is how transmissible a disease is. So different diseases have different betas. You know, measles, which is really infectious, might have a super high beta, whereas something that's less infectious, you know, even something like HIV might have a lower beta. Gamma represents the rate at which people naturally recover from the disease. So it could be something like, let's just use an example of like 6 months, or 1/6 months is actually how it's written. So the gamma for trachoma might be 1/6 months, and that represents how they go from becoming infected to becoming susceptible again. Okay, yeah?
Sorry, I already have a question.
Yeah.
This is the simplest one.
Does data also include something about how much of the disease is in the population? Not
So
just
that's,
about infection?
yeah, so, I mean, you're natural. So what actually should be, what actually I should have written here, what I did to make it simple was beta times I, which is the number of infected people in the community, times S, because what you want to do is take the combination of how many possible interactions any infected or any susceptible person in the population can have, and then multiply it by like a transmission coefficient. So that's great. It kind of leads me to the very next thing that I am going to show, which is, I know this is a differential equation representing infection, but I'm going to explain it in a really simple way. Basically, this equation right here tells you how many infected people you have in your community at any given time point, okay? This is what it looks like when you graph it. On the y-axis, you have number of infected people in the community, and on the x-axis, you have time. So I started this equation by assuming that there's 2 infected people in the community, and then I just looked to see what happens over time. So it starts off with like 2 infected people, and then it exponentially grows, and then at some point it saturates, and then it reaches this endemic equilibrium. So the endemic equilibrium is at 50. And this is what's called a deterministic model. So this equation is deterministic, meaning if you give it some initial starting condition, i.e. 2 infected people initially, it's always going to give you the same result, okay? And if this were the case in real life, if this is how infection behaved in the real world, man, I'd be done and published in like 2 weeks. But this is not what happens. This is what happens in real life. This is what's called a stochastic model. And as you can see, it's like really jagged. It kind of looks somewhat like that, but not really. And this is because in real life, you have chance variability at any given time point. There's a chance that someone could become infected, or a chance that someone could become, who's infected becomes susceptible again. And you have to kind of account for that. It's kind of the same principle that happens in modeling stock prices. There's just random chance variability, and you have to account for it. And here I just overlaid 2 stochastic graphs with the deterministic. And you can see that the stochastic models kind of approximate the deterministic one. And the green model is, actually I just ran the same exact thing twice, and you can see you get two completely different results, the green and the blue curves. And that's what happens when you throw chance into the mix. So now I took my very simple model and I added a stochastic effect. I'm going to make it a little more complicated and add antibiotic treatments. And then I'm going to add outside infection, and then I'm going to add what's called a random effect, which is different villages can have different levels of beta or gamma, like they can have different transmission coefficients. So I will allow for that. Yeah.
I'm sorry, does beta change depending on how many people are infected?
It doesn't change based on how many people are infected. It changes based on the conditions which propagate transmission. So things like how close people are in contact in one particular village might affect what the level of beta is. The number of people in the village is independent of, like beta is an independent variable from that. If that makes sense. I think I understand what you're saying. Like, if you have more people that are infected in a village, like you're going to increase the number of people who become infected over time, right?
Right.
Is that kind of what you're getting at?
Mm-hmm.
That's accounted for in the equation, independent of beta. Yeah. Okay, so now we have a model, and we have data, prevalence data. So how do I figure out what are the values for my parameters, beta and gamma, and this efficacy that I'm trying to model? It's through these equations. I won't try to explain them. I'm just going to tell you what they allow me to figure out. These are called the Kolmogorov-Ford equations, and what they basically say is, let's say you have 100 people in your village. This turns into 101 equations. It tells you what's the probability that I have 0 infections in my village at some time. What's the probability I have 1 infection, 2 infections, 3 infections, all the way up to 100. So I'll use that to do the following. I'm just going to give you an example so this makes a little more sense. I'm going to be doing what's called maximum likelihood estimation. So imagine the left 2 columns are the data I have for some village. I have a baseline value of 50 infected people. At 6 months I have 20 infected people. At 12 months I have 30 infected people. That's the data that's given to me, okay? What I do then is take the equation on the previous page and ask the following question. Let's look at the 3rd column. So assume if I, for example, have a beta equal to 1, a gamma equal to 0.5, and an efficacy of 85%. These are values that I made up, and I'm trying to figure out if those are correct values. The equation on the previous page will allow me to say, what is the probability with those parameter values that I saw 50 infected people at baseline? Well, the probability is 12%, 0.12. What's the probability that there were 20 infections at 6 months? Well, the probability was 45%. And what was the probability that I saw 30 infections at 12 months? It's 34%. That's what I see with the first set of parameter values. With the second set of parameter values, which is the 4th column, I kept the same beta, same gamma, but I changed the efficacy to 65%. What it's saying is, with those values, what was the probability that I saw 50 infections at baseline? Well, it was 56%. 67%, 76%. Those are a lot better than the first set of parameters. So I'm getting closer. I'm getting closer to the correct true values of the parameters. And now you use a set of complex algorithms to figure out what is the actual optimal value for the parameters that you're looking for. And remember, the parameters that I'm solving for are beta, which is the transmission coefficient, gamma, and then efficacy, which is the true answer that I'm looking for. So that is basically what I'm going to be doing. So I have periodic cross-sectional RCT data from 24 communities in Niger and communities in Ethiopia as well, who received annual mass treatments of antibiotics each year for 3 years. The infection is determined by PCR swabs of the conjunctiva, and I'm going to be creating stochastic SIS models using maximum likelihood estimation to solve for optimal values of beta, gamma, and efficacy. And then I can figure out what the confidence interval for my efficacy is by bootstrap resampling the data. And that is how I'm going to come up with my answer of what the efficacy is of these antibiotics. And it's an important question to talk about, because if the antibiotics are losing efficacy over time, i.e. there's resistance that's developing, well, this whole global elimination program that the WHO is trying to implement is not going to work, or it's going to work on a time scale that's 10, 15 years longer than they thought. Yeah.
How was the 3-year mark chosen?
The trial just ran for 36 months. So it's, I think Tom might know more than I do about that because that's something that was developed before I showed up.
NIH grants are 5 years, and it takes a year to start and a year to analyze.
So you're
So
hoping
you do
that
a
if there's
3-year study.
if there's resistance developing, you're hoping that you'll see it within the first 3 years of the implementation.
That's a very good point. I'll let it get back to Salman, but the trials actually were comparing different treatments. So this is a secondary outcome from the trial. And Salman will have some data from Niger in 3 years. He'll actually have data in Ethiopia after 7 or 8 years as well.
So this is kind of what the data looks like, which I showed you earlier. This is different data than what I'll be analyzing from Niger. This is from Ethiopia, and they received biannual treatment, so every 6 months for 4 years. And the green arrows represent each treatment. But I just put it here to show you guys that what I'll be doing is I'll be calculating the efficacy for year 1, year 2, year 3, and then I can see, well, is the efficacy changing over time? And actually, secondarily, I mentioned a little bit earlier that I was also testing loss of immunity. What that means is I'll be testing to see whether the beta stays the same or changes over those 3 years. So these are kind of the project aims that I had. Loss of efficacy, I'll compare the three years using ANOVA analysis. I can calculate change of transmission. And then later in the year, if I have time, I'm going to be trying to look at whether we can use these models to predict prevalence of trachoma, which the WHO is becoming more interested in. And no one's really doing prediction. So currently where I am with my project is learning the math, which as you guys saw is not easy. So that's kind of where I am. I'm building the model, learning the mathematics required to understand those models, and then hopefully soon I'll be fitting the data that we have to the models. And then, like I mentioned, in a couple weeks I'm going to Ethiopia and learning kind of how the data is being collected and actually doing some of the data collection myself with the swabs. So that should be pretty fun. So lastly, I just want to thank Tom. He's been giving me a lot of guidance, and some other people in the lab, Catherine Ray and Feng Shen, who really helped me understand the mathematics of what's going on. I've been working with the modeling group, and the Proctor Foundation has been extremely generous in helping me fund my travel, so extremely grateful for them. Yeah. So that's my talk. If you guys have any questions, I'd like to open it up.